Hello, my name is Scott Requadt. I'm the Chief Executive Officer of Talaris Therapeutics, and I'm excited to share our story with you. So there's no question that the advent of cell therapy has revolutionized how we think about treating complex diseases involving multiple biological pathways, most notably cancer. At Talaris, our goal is to harness the great power of cell therapy to fundamentally change how we think about and treat disorders of the immune system, where the immune system undesirably attacks good tissues, and where to date the only treatment option has been to suppress the immune system rather than to change the underlying pathophysiology of the disease. The two biggest examples of this are organ transplant, where an unfettered immune system will attack and reject a life-saving transplanted organ, and autoimmune disease, where the immune system, for whatever reason, attacks some aspect of self. Our therapy has the potential to address the root cause of this problem, enabling a patient's own immune system to treat a transplanted organ itself and potentially to restore tolerance to self in somebody who suffers from an autoimmune disorder. Our lead indications are in the organ transplant space, where we have conducted a compelling phase 2 clinical study and have now started a phase 3 registration study in living donor kidney transplant, and where we also have an active development program to extend our technology to using cells from deceased donors for not only kidney, but other organ transplant. In the autoimmune space, the FDA has recently given us the go-ahead to initiate a phase 2 study in scleroderma, a multi-system autoimmune disorder currently very poorly addressed by existing therapies, where we believe our therapy has the potential to try to restore immune tolerance to self. And this could open the door to a number of additional severe and refractory autoimmune disorders that could be amenable to our therapy. Our lead product, which we call FCR001, is a single-dose cell therapy. It comprises both stem cells and immune cells, which we harvest from a healthy donor and which we administer to the patient with a non-myeloablative conditioning regimen to create enough room in their bone marrow niche to allow them to receive the stem cells from the healthy donor. It builds on a Nobel Prize-winning discovery, which for 60 years has eluded being reproduced in humans. But using this basic biology and our therapy, we've generated a very compelling phase 2 data set in our lead indication, which is living donor kidney transplant patients. And on the basis of that pretty compelling data set, we received RMAT designation from the FDA. We recently started a phase 3 registration study in our lead indication, living donor kidney transplant, and we have plans to initiate 2 more studies next year to demonstrate the potential of our product to really represent a pipeline in a product. We do all of our cell processing in-house using a relatively simple but proprietary set of processes, and therefore it's a highly de-risked process compared to many other cell and gene therapy companies. We raised $100 million in a Series A financing back in November of 2018, and we have additional plans that ensure that we're going to be very well financed to pursue an aggressive value creation plan in the years to come. Starting with the unmet need, in the organ transplant space, while immunosuppressive agents are life-saving because they prevent your body from rejecting the donated organ, ironically, those drugs are also inherently toxic to the kidney, and they lead to an inexorable decline in kidney function over time. As a result, the average life of a transplanted kidney is typically no more than 15 years, and younger kidney transplant recipients need to receive multiple kidney transplants over the course of their lifetime. Another issue associated with these drugs is that there are a number of cardiovascular comorbidities associated with them. And of course, since you're chronically immunosuppressed, you're at significant risk of serious infection, COVID-19 being a perfect storm for organ transplant recipients who are on chronic immunosuppression. And given what we now understand about the role of the immune system in fighting certain cancers, it's not surprising that studies have shown that organ transplant recipients on chronic immunosuppression are at significantly higher cancer risk. Not to mention high pill burden, quality of life issues, leads to people not complying with their medication and premature loss of their kidney transplants. If we could get rid of all of these toxicities and comorbidities by getting rid of the drugs that cause them, this would be a complete paradigm shift in the organ transplant space. Our phase 2 study, which was conducted between 2009 and 2016, was in 37 living donor kidney transplant patients who received a kidney transplant as well as our therapy. Of those patients, 26 out of 37, 70%, we were able to wean off all immunosuppression within 12 months of their therapy. We've now followed this entire cohort for a median of over 5 years and the longest out to 10, and 100% of these patients who came off immunosuppression have been able to stay off immunosuppression. In a subset of these patients who had a pre-existing autoimmune condition that attacked their kidney and led to their kidney failure, we were able to tolerize a number of these patients. And what we noticed is that of those patients who had a pre-existing autoimmune condition that attacked their kidney and where we were able to tolerize them to their donated organ, not a single one of these 7 patients had recurrence of their underlying autoimmune disease, notwithstanding that you would typically expect that to happen in anywhere between 1/4 and 60% of these patients. We believe that this points to potential proof of concept that our therapy can be used in the autoimmune space in addition to the organ transplant space. The last point I'll make about this slide is that we learned from some of the earliest patients that were dosed, and we optimized aspects of our protocol, of our cell therapy, of our doses, of our conditioning. And when we locked all of these down in the latter half of the trial, of the last 17 patients that were dosed in our trial, 14 of those 17 patients we were able to get off all immunosuppression. And these are the doses, this is the product, these are the protocols, and these are the patient inclusion and exclusion criteria that we're carrying forward into phase 3. One of the things that's notable about taking stem cells and immune cells from one patient and putting them into another is that typically you need to see a very high degree of immune system match between the donor and the recipient. The poorer the match, the less the likelihood that you'll get good engraftment of the stem cells from the donor, and the higher the risk of graft versus host disease. So typically you want to see as much as a 6 out of 6 HLA match or immune system match between donor and recipient. Remarkably, in our trial, almost 3/4 of the patients that we were able to get off all immunosuppression had very poor degrees of HLA match between donor and recipient, typically only 1 or 2 out of 6 HLA match. And yet HLA match or mismatch did not correlate in any way with safety or efficacy of our outcomes. The implication being that if somebody is an organ donation match, then the fact that their immune system might be quite profoundly mismatched from the organ transplant recipient would not prevent us from using that individual's stem and immune cells as the basis for our product. We followed the phase 2 patients, as I mentioned, for a median of over 5 years, and the safety profile has been very comparable to standard of care kidney transplant, both in terms of kidney as well as patient survival, and very comparable with what you would expect in HLA matched stem cell transplant, notwithstanding that a very significant proportion of our patients were profoundly HLA mismatched with their donor. The other hypothesis that one would have is that if one were to remove the immunosuppressive agents, then you would not have the toxicities associated with them. And we have seen that in our phase 2, as we follow these patients over time, we see good preserved kidney function in our tolerized patients, whereas in a comparable cohort of patients who were concurrently transplanted at the same site who met all of our phase 2 eligibility criteria, we have seen a steady decline in kidney function over the first 5 years, which we would expect will continue to be exacerbated in the future years. Moreover, you would expect to see much less dependence on cardiovascular comorbidity medications such as antihypertensives or high cholesterol medication, and that's exactly what we've seen in our tolerized patients as compared to what's been seen in that concurrent cohort that was transplanted in the same study. Our therapy itself consists of 3 active ingredients, one of which are CD34 cells, another of which is alpha beta T cells, both harvested from the donor, but the critical ingredient is facilitating cells. This is a unique type of non-stem cell that was discovered by our founder, Suzanne Requadt, who's a transplant surgeon and immunologist by trade. And what she had discovered that these cells, which are CD8 positive and yet T-cell receptor negative, is that they have unique functions insofar as they promote stem cell engraftment in unmatched recipients. They also appear to significantly prevent graft versus host disease in mouse models and upregulate a number of tolerogenic pathways. These cells are critical in terms of enabling the reconstituting donor-derived immune system to tolerate the host and also the reconstituting recipient immune system to tolerate the donated organ. On the basis of our compelling phase 2 data, we've now initiated a single registration study we call Freedom 1. It will be in 120 adult living donor kidney transplant patients, of which 80 will receive a kidney transplant and our therapy, and 40 will receive just a standard kidney transplant. It's an open label study, and the primary endpoint of this study is what percent of the 80 individuals who receive our therapy are free from all immunosuppression and have not had a rejection event 2 years after their transplant. We do have a surrogate marker, which is a simple blood test 6 months post-transplant that essentially predicts to the clinician which patients can be safely removed off immunosuppression. And that marker has in fact proven to be 100% predictive of the ability to get someone off immunosuppression, and everyone who came off immunosuppression stayed off. And therefore this surrogate marker is highly predictive of both weaning off immunosuppression as well as long-term durability. We're going to be enrolling this study at 15 sites across the United States, and we have dosed our first patients. As I mentioned earlier, unlike many cell and gene therapy companies, our manufacturing process is relatively simple. It can be completed in less than 24 hours. We're not doing any genetic engineering of the cells. We're not doing any ex vivo expansion of the cells. We do all of the processing entirely in-house in our state-of-the-art cGMP cleanroom facility, and we have existing capacity to supply all of our clinical and commercial early commercial needs. Very importantly, and unlike many cell and gene therapy companies, we're trying to transition an academic process into something that can be robust enough for commercial use. We are not having to change any aspect of our manufacturing controls or processes between what was so successful in phase 2 and what we'll be using in phase 3. Moreover, we have broad issued composition matter claims covering our cell doses and a number of additional pending claims that would run until 2038. We do believe that our therapy has the potential to be a pipeline and a product. 12 months from now, we envision having 3 clinical studies underway: our Freedom 1 registration study and our lead indication, our Freedom 2 study, in which we'll go into living donor kidney transplant patients who previously received a kidney transplant. We'll go back to the donor and harvest cells and see if we can induce tolerance after the fact. And in scleroderma patients, where we'll be using donated cells from a healthy donor in order to try to induce or restore tolerance to self in these patients. We also have an active preclinical program to show we can use the exact same cells that we harvest from a living donor from a deceased donor. And that, of course, would open up the remaining 75 to 80% of organ transplants, which are from deceased donors. Our therapy addresses a huge market. Our lead indication, which is in living donor kidney transplant, there are about 6,500 of these a year, and that's what we're targeting with our phase 3. Our phase 2 in delayed tolerance would open up a part of the prevalent population of people who previously received living donor kidney transplant, and our deceased donor program would open up twice as many as the living donor program. And our scleroderma indication has an incidence of 5,000 and a prevalence of roughly 20,000. So a significant initial patient population for us to address, and we see the opportunity for a number of fast follower indications, all of which could be accessed with the same product, made in the same way, dosed in the same way. We have a very experienced board and senior officers. Nancy Krieger, my chief medical officer, also a transplant surgeon by training, has been working in the transplant space for over 20 years. In summary, Talaris has very uniquely identified a cell therapy that we believe has the potential to induce durable drug-free immune tolerance in two areas of great unmet need, organ transplant, as well as severe autoimmune disease. Our lead program is in a phase 3 registration study for living donor kidney transplant, and we'll be kicking off 2 more studies next year to demonstrate the much broader potential of our product. We do all of our cell processing in-house, and it's largely a de-risked and locked-down process. And because our trials are open label, we anticipate significant data flow from our studies over the course of the next 18 to 24 months. Thanks so much for letting me introduce you to Talaris.